Okay, thank you, Mark, and thank you everybody for joining today's webinar. We're going to go ahead and get started. So today is April 18th. And just to give you a little bit of history about myself, I am going to MC today's session. And so just want to give you a little bit of background about myself, and then we're going to get introduced to a whole bunch of other panelists that are going to be helping us do some Q&A at the end of the webinar today. So for myself, I have been really fascinated with natural systems all my life. I grew up on a homestead as a child. My parents, we raised our own, fruits and vegetables and raised our own animals. And so, growing food and kind of being in that environment is very, natural to me. And as I got older, I'm also a computer nerd, so I went into the high tech industry and did that as kind of most of my career. And as partway into this, my wife and I bought our own homestead. We bought 5 acres here in Northern California, and I really wanted to, um, grow our own food and do the things I did when I was a kid. But my parents, they ran it in a very conventional sense or chemical sense. We fertilized, we rototilled, we, applied the different pesticides and herbicides and fungicides and all that kind of stuff. And so that's what I knew growing up. That's what I did when I got, our homestead. And I just wasn't happy, A, with putting chemicals out on the food I was eating, but two, I was having to put more and more of those inputs, and I was getting more pest problems, and the quality of, the production was going down, and just was not very happy with that. And I started to research, say, hey, there's got to be a better way to do this. And I first came across permaculture, kind of the design science and growing food forests and stacking functions and all those kind of good things, and started to make a change here on our homestead. But I understood what I was doing, as far as the design science, but I didn't understand why. Why was the plants responding so much better? Why was I getting more production? And I wasn't using all these chemical inputs. And I read a book called Teaming with Microbes from Jeff Lowenfels. And in there, Dr. Elaine Ingham writes the foreword. I didn't have to get past the foreword. I read the foreword and I was, wow, okay, this really starts to answer a lot of questions for me. And I went out and did a search on Elaine and found some YouTube videos. I was like, okay, this is something I need to know more. I've got to be able to sink myself into this. And so, the homestead, it was a hobby, became a very big passion. And then I decided, you know what, I've spent enough time in the high tech industry. I was kind of done with the corporate life, the corporate world. I wanted to do something different. And that's when I made that transition. And I'm so very happy I did. I ended up becoming a certified soil biology consultant. I created my own company, and now I do this full time. I help farmers convert from, the chemical paradigm that they're kind of tied to, and to get them to switch to biological farming. So I'm very, very happy and blessed that I actually did make that change. Okay, so enough about myself. Um, let's go ahead and talk about what we're going to talk about today. And we're going to introduce some folks that are going to be our panelists, and they're going to be, folks that are very familiar with being a lab tech, running a soil biology lab, and that's going to be the main thrust of what we're going to be talking about today. But we're also going to talk about the soil web training program in general. So what we have the foundation courses, or what they call the FC courses, and this gets into the theory and the science. And then we have the certified or the consultants training program. And this is to help train you to become a consultant, so you can do the kind of work that I do, helping farmers or government organizations or parks and rec and golf courses. I mean, it's pretty limitless as far as the amount of people that you actually can help. And then we're also introducing a new program called the Certified Lab Program. So this kind of fits between, just knowing about the foundation courses or learning the foundation courses and the consultants. This is where you're able to run just a lab where you're doing soil biology assessments. And we'll talk about that. And then we're going to have an introduction from Elaine, and so she's going to talk about her background and her history and how she got where she's at. And then we're going to end, and probably the vast majority of the webinar today is going to be around Q&A. So we want to be able to hear from you folks what kind of questions that you have. And if you can, post your questions on the dashboard. There's a questions box. Go ahead and start posting your questions. And we have moderators in the background kind of selecting questions for review. And then I'll be bringing questions up for our panelists, and we'll have discussions with Elaine and so forth. Okay, so that's what we're covering today. The webinar rules of engagement. So everybody that's not a panelist or organizer has been put into muted mode, and that's just because we have, expect a lot of people to show up and don't want to have the background noise. And like I mentioned before, if you have a question, please click the question button and start. You can start submitting questions now if you want to, or as we start going to the discussion, go ahead and pop them in there. Okay, so let's talk about our panelists today. So, consultants, folks of us that are certified consultants, they're doing the consultancy work. I'll be representing that as well as Tanya. And then we've got compost producers, Keisha and Casey. They have Catalyst Bioamendment out here in Northern California. Big fan. I use their products all the time. And then we have Soil Food Web certified lab technicians. So Wes and Nancy will be representing the lab technician portion of this. And I just want to point out that all the folks that are on the panel today are, part of the, we have the skills. We've been certified in the lab tech aspect of doing that work. So you'll get to hear a lot more from the panelists as we go through the Q&A sections, but just kind of know the different parts that we're trying to represent here. Okay, so let's talk about the current Soil Food Web training program. And then we're going to add in the CLP, which is a certified lab program. So starting with the foundation courses, and this is where you start. This is where you get to learn all the different theory and the science behind the Soil Food Web approach. It's very important before you actually start doing the practical aspects of this work that you know the relationship between soil biology and soil health and plant health, and the interaction between the soil biology and the plants. And this is where Elaine goes through and really gets into the science. And I don't want to scare anybody off by thinking I've got to have a master's degree in chemistry to be able to understand this stuff. No, Elaine makes it very accessible for folks, and builds upon that knowledge as she goes through those courses. So feel free, if you don't have a science background, doesn't mean you can't do this. The other thing too, we live in the age of the internet, so if there's certain terms that might come up, you can always pause the training, go look up the word, get more information about the definition, and then go back into it. But Elaine, usually, if there's some kind of word that's not very common, she's going to go through and give you that explanation what that actually means. Okay, so once you've taken the foundation courses, then you're going to move into the consultants training program. And this is really where you get to put all that theory and science that you learned into practice. And when we go through the consultants training program, you're going to prove out that you can make good biologically complete compost, and you can make good biologically complete compost teas and compost extracts. And then you're going to have a project where you're showing that you can apply all those skills that you've learned to some either farm or yard or whatever that project is that you define, that you're able to change the biological profile to match the plants that you're trying to grow. And at the end of that, that's when you can become a certified Soil Food Web consultant. And now you can go out and work with the different farmers. You get a listing on the website and so forth. And we'll talk a little bit more about that as we get through the presentation here. Okay, so what's new? We're adding in the certified lab program. And so this kind of fits between the foundation courses and the consultants training program. So, for some folks, they just take the foundation courses, they want the knowledge, and they may run their own farm and say, okay, that's great, good enough. Other folks say, hey, you know what, I want to do this full time. I want to be a consultant. I want to really help push people in that direction. And others really like to just do lab work, where you're under the microscope and you're seeing all these neat organisms and critters and doing assessments and so forth. And so we want to make sure there was a program that kind of fit for that. And you can also just go foundation courses first and do a lab, run your lab for a few years, and then decide, maybe I want to go do the consultant training program, become a consultant. So there's a lot of options in that sense. Okay, so let's talk about the offer that we have today. So foundation courses typically cost $5,000, and the certified lab program on its own costs $1,000. And the offer that we're putting together is for $3,600 for both of those two classes. So that's about a $2,400 savings. And I'm going to give you a link a little bit later in the presentation so you can go online, get more detail about the program, and that's where you can actually go through and register. So yeah, it's a fantastic deal that the school's been able to put together. All right, so let's talk about a little bit more into, what do you learn in the foundation courses? We talked about, hey, you're going to learn about the soil food web and the interaction between soil biology and plants, but let's get down a little bit more. So we put the foundation courses into 4 different groups, and we call them foundation course 1, 2, 3, and 4. And foundation course 1, which is the biggest block of training that you're going to go through, really is the, going through the theory and the science. And so you're going to learn about what is the soil food web, what makes up the soil food web, what are all the different microorganisms and what are their functions. And then we'll get into how do you work with the soil food web as far as the different products that you use and the practices that you might apply. And then we also get into a lot of case studies, and the case studies are great because it's not just in agricultural, but it's parks and recs and golf courses and other things. So it does really show that the breadth of the opportunities, and where we can make a really big difference with changing folks into working with soil biology or having soil biology in the forefront of their mind when they're actually doing that type of work. And then we move into Foundation Course 2, which is making biocomplete compost. So this is going to be kind of teaching a little bit around the practical aspects of, okay, now we understand about the soil food web and how important soil biology is. What are the tools that we have to be able to make an impact? And it starts really with compost. And so we'll talk about, what is biocomplete compost? How do you make it, both on a small scale and a large scale? And we'll get into the difference of thermophilic composting versus static composting versus vermicomposting. And then, once you've made all this wonderful compost, then how do you actually apply it? The physical form of compost itself. And then Foundation Course 3 is making biocomplete extracts and tea. So once you've made really good biocomplete compost, now you might want to have some liquid amendments, another tool that you can use to be able to apply it. And so we teach you the difference. What is an extract and what is a tea? How do you make an extract? How do you make a tea? And then what are the application methods? It's a foliar spray, or if it's a soil drench, or if we're doing a deep root zone injection and things like that. And then we get into Foundation Course 4, which is the microscopy. And as a tool that we use, probably one of the most important tools that we use from the soil biology standpoint is a microscope. It's not like you're going to be able to reach in the dirt and pull it up and go, "Ooh, look at that nematode," or "Look at that amoeba," or "Look at those different organisms." They're microscopic. So the microscope really helps us be able to do that identification. And many of you may not have experience with a microscope, so we'll break it down. What is a microscope? What are all the different parts and pieces of a microscope? How do you actually adjust and control your microscope? And then we'll get into the how do you do identification? Because when you're looking through there, you're going to see probably a bunch of organisms and objects you've never seen before. So what is an amoeba? What is a flagellate? What is a nematode and a fungal hyphae and things like that? And then at the end of this, you're looking for data. You want to be able to say, I was able to observe these things, and I want to be able to classify it and say, all right, it had so much bacterial biomass or fungal biomasses, and what are the ratios? And so there's a lot of data that we collect, but we also then need to interpret that data. What does that data actually mean? So we get into the interpretation aspect of it. Okay, so this is, if you're going just to do the certified lab program, you need to know all these things going into that. And so then we move on to what is in this certified lab program. So you finished your foundation courses. Now you're going to sign up and you're going to be assigned a mentor, and you're going to work on these one-on-one sessions with a mentor. And the mentor is going to give you things to practice. Okay, based on our last session, go look at these assessments, try to identify these things, take pictures and things like that. And you're going to build your confidence, your skill as you go through that process. At the end of this, you're going to take an assessment. We want to make sure that you have the skills to be able to A, use your microscope, and then B, be able to do the identification, so that you can determine, yeah, okay, that was an amoeba or that was a flagellate. So we designed the program that it can be done in as quick as 6 weeks. And if you did this full time, where you did the foundation courses and did your CLP, you could complete this in a 6-week period of time. But we know for some people that's not as fast as they want to go, or they have other things that you may be working on. So we also then offer to make sure that you can complete the foundation courses in 1 to 2 months. So you don't have to do it in 6 weeks. You can do it, the foundation courses, in a 12-month period, and then another 3 months to complete the certified lab program. So you have a lot of flexibility as far as to work this in your own time frame that works best for you. The other thing you're going to need to become a certified lab is your lab equipment. And so you do need to have a microscope with a camera. And there's a certain type of microscope that we would, help you spec out to say, okay, it needs to meet these specifications. And then once you have that, you could, purchase a microscope and a camera. And you're also going to need your lab equipment. So test tubes and slides and cover slips and pipettes and things like that. And we break it down. We give you a list of all the different things that you'll need to stand up a lab. And, you can budget this for as little as $300 to $600 on your equipment. And with microscopes, this is going to be your biggest expenditure You can start with a budget-friendly microscope, and you can definitely, move your way up into a much more expensive microscope. And, personally, I still use the microscope that I started with. I think I spent $500 on my microscope, and it works really good. The more expensive microscopes, they look a little bit sharper, they're a little bit better, and if you're doing a lot of assessments, yeah, you're going to probably get better efficiencies and a little bit less eye strain and things like that. So you can step up to a more expensive microscope if you want to. All right, so now once you pass this assessment, the other advantage you get is you get listed on the Soil Food Web School's website. And so when folks go to the website and they're looking for a lab to be able to submit their results, you'll be listed there with all the other labs. And a lot of people will look for somebody who's close regionally, and so it's a good way to be able to market yourself through the website. The other thing too is there's a Launch Your Lab guidebook, which really is some instructions on how to work with farmers, how to do that kind of engagement aspect, and then also how to set up or stand up your small business as far as the lab is concerned. So we try to give you as much resources as you possibly can. And one other thing too is you do get access to a forum where there's a community of folks, so you can ask questions and post pictures for identification and things like that. So there's a pretty good support community and system behind all of this as well. All right, so if you do want to know more and either register or sign up or just get some more details about the program, we do have a URL down here at the bottom. It's www.soilfoodweb.com, and it's LYL, Launch Your Lab. So it's a fairly easy URL to be able to get access to. Okay, so I'm going to now hand this over to Dr. Elaine Ingham so she can talk about where she's been, her history, how she's got here. And Elaine, I'm going to hand this over to you.
Okay, thank you, Brian. Thanks for going through all of that material. Hope that people are getting interested. I, of course, have been working with soil biology through all of my life. I think back to the time when I was 6 years old, and my father, who was a professor at the University of Minnesota in veterinary medicine, didn't know what to do with me, was kind of babysitting me, and so took me into the lab with him. And got to the lab and plunked me in front of a microscope and taught me how to focus it and how to get a good image, put a couple of petri dishes that contained interesting pathogenic organisms. So got a little instruction on not to drink any of that water. Put drops on the microscope slide and told me to count the E. coli in all of those samples, and while he went off and did this other work. So I think I was somewhat predestined to go into something where we use microscopes to be able to measure what's going on in the soil. If you can't measure things, you can't manage them. And therefore, when we've determined, so through my education, we've shown what the interactions between these organisms and the plants actually are and how much that benefits the plant. You come to understand and realize why you need that microscope to make certain that the organisms that are needed are present in your soil, present on the surfaces of the plants. So I went to St. Olaf College back in Northfield, Minnesota, and yes, you can hear the Minnesota girl in the every once in a while, yeah, for sure. Got a double major in biology and chemistry from St. Olaf College. It's one of the premier pre-med students go there because we have the highest rate of those students who want to go into medicine being accepted at various institutions around the country. So I was going to go to medical school. That was my plan until I worked for the summer in the heart hospital at the local university and realized that the attitude of the doctors was not my reason for doing anything. I'm not focused on money. I want to make the world a better place. I want to help us stop using pesticides and inorganic fertilizers and all the toxic, highly disturbing practices. How are we going to do that what's the mechanism and understanding that mechanism. So instead of going to medical school, I went to Texas A&M graduate school for my master's degree in marine microbiology, which maybe seems a long way from soil, but the overarching principles are the same. When we're looking at microorganisms, the things that they do are the same regardless of whether we're in strictly terrestrial soil situation or whether we're in sediments in a spring or a river, lakes, things like rivers, streams, or the bottom of the ocean. So any place you go, you can still apply these overarching principles. What changes is the specific conditions, the specific way you apply that overarching principle. So in marine microbiology, I discovered for the first time that the existing scientific methods for understanding any of the ecosystems that we deal with was completely inappropriate. The methods that had been developed were all developed to be able to deal with problem organisms, to deal with diseases and pests and the really bad guys, if you will. We knew a lot about the habitats of the bad guys, but we knew nothing about the habitats of the good guys. And so demonstrated that by working with oysters and finding out why the oysters in Galveston Bay down in Texas weren't growing and why it wasn't a commercially viable situation in Galveston Bay to be raising oysters. And it was all about the biology. I was the person who discovered that oysters have to have these certain species of bacteria in their digestive system in order to help them convert whatever they were grinding up, whatever they were taking in, had to be massaged, had to be dealt with, decomposed to some extent by these bacteria before the oyster could take up those food sources. And so oysters that didn't have those microorganisms were not going to grow very well. They weren't going to grow at all and would ultimately die, or they would grow only very slowly, which is not a story for success when you're trying to sell oysters into restaurants. So from Texas A&M, I went to Colorado State University in terrestrial sciences, so soil microbiology, to work on my PhD. And as soon as I arrived, my major professor, Dr. Don Klein, told me to go around and talk to all the people in the soils departments, any place where they grew plants, go and talk to them and find out what they think about biology in the soil. What is it there for? What's it's doing? How important is it? And at that time, the attitude by anyone in the world of growing plants or soils, their attitude was that the bacteria, fungi, protozoa, nematodes, microarthropods, the earthworms, they were just there. Don't worry about them. They're not important. They don't in any way influence the growth of plants. And my response to that was, well, but these organisms have been on this planet for the last, well, somewhere between 3 and 4 billion years. Mother Nature doesn't put up with something that isn't doing something that is beneficial in terms of growing plants. She's not going to put up with it. She's not going to support that loss of energy. If these organisms really had no role and no function, they wouldn't be there. So it's really up to us to try to figure out what they're doing in these. Are they good? Are they bad? Are they just kind of maintaining status quo? What's going on? So working with the project that my major professor gave me, which was to figure out a way to measure bacteria and fungi and protozoa and nematodes and figure out which ones were active and which ones were not active. That was my project for my PhD, working on how do you measure activity of these organisms. And of course, microscopes were the way to do that. kind of had established that very clearly back working on oysters. You would see hundreds of different species of bacteria and fungi and protozoa in the digestive tract of the oysters, but the classical ways to assess numbers of organisms and diversity of organisms would tell you that there was only 2 or 3 species, when in fact there were hundreds of species. The methods that were being used were just completely inappropriate. So how are you going to find out how much fungal biomass, how much bacterial, et cetera? What was the activity of those organisms? That's the work that I did for my PhD, coming up with these methods that you don't have to do a different way of sampling in order to get an understanding of the full food web. And then, what are these organisms in the soil to do? What is their point and purpose? So the major paper, probably the most referenced paper in the world of soil ecology, is the one that came out of my husband and my PhD work, where we showed that as we increased the diversity of organism groups within the food web, directly correlated with the uptake of nutrients into wheat plants. And so that extensive work where we were showing as the biology increased, as nutrient cycling was performed by these organisms in the soil, we were essentially using the plants to document the improvement in soluble nutrients. And this is, if you take that logical next step, where do soluble nutrients come from in soil? The soils people would always tell you that you have a very poor soil. They would do their soil chemistry, looking at soluble nutrients in your soil. And anytime that dropped below a certain particular level of concentration of the soluble nutrient, then they'd tell you that you had to put in these inorganic fertilizers. You know, the classical green revolution approach to dealing with plant production. And you start thinking about an old growth forest. Nobody's out there in an old growth forest putting out inorganic fertilizers ever. Those old growth forests have grown, and every year those old growth trees are putting on another ring of wood around their outsides, around all of the woody parts. They're increasing in diameter, slowly but surely. But you think about an old growth forest tree that's maybe 200, 300 feet tall. How much nutrient is being sequestered and stored in that tree? And that nutrient isn't coming back into the nutrient cycling in the soil anytime in the near future. Some 2,000 years from now, or 1,000 years from now, maybe that nutrient will get back into the soil system. So where do the nutrients come from to feed that old growth tree next week, or a month from now, or 2 years from now, or 100 years from now? Where does it come from? You can't just be looking at the soluble nutrients in the soil. So that led me to go to the soil chemistry literature and look at how much total nutrient was present in the soil, because that's where the biology in the study that we did, that's where those nutrients were coming from. They were from the structures in the soil, the crystalline structure, from the sand, the silt, the clay, the rocks, the pebbles. Every boulder, every grain of sand, every rock out there in your ecosystems, in your soils, are covered with bacteria and fungi happily decomposing all of those materials, pulling the nutrients out of the sand, silt, clays, rocks, and pebbles and into the bodies of the microorganisms. So bacteria and fungi are now elevated in their nutrients. There is no organism on this planet that has a higher concentration of all nutrients than bacteria. Fungi come in second. So they're concentrating, they're holding, they're preventing loss of those nutrients. So we can have that storage right around the root systems of the plant, because the plant is putting out exudates to inform those bacteria and fungi what nutrients the plant requires and what enzymes those bacteria and fungi should be making in order to pull those nutrients out of the sand, silt, clays, rocks, pebbles, and everything else. And so the plant's really in control of building up this reserve, the storage of the nutrients right around the surface of that root. So then when protozoa, when nematodes, when other predators come along and eat the bacteria and fungi, the concentration of nutrients is so high in the bacteria and fungi as compared to their predators that the predators have to release those nutrients in a plant available form, in a soluble form. That's where soluble nutrients come from in your soil. If you're having a problem with fertility, the problem probably actually is not that you don't have the nutrients in the soil. You do too. There is no soil on this planet that lacks nutrients to grow plants for the next 100,000 million years, because those organisms can break down the rocks, the pebbles, the parent material. That's all going to get solubilized and turned into bacteria and fungi, which will release those soluble nutrients when they get eaten. And that's what your plant's going to take up. So where the highest concentration of bacteria and fungi are is where the plant's feeding them, right around the root system. So that right around the root system is where the highest concentration of those plant available nutrients are. But it took doing these kinds of experiments, looking and understanding what the actual function was, to understand that we've been doing absolutely the wrong thing in the Green Revolution. It's not a green revolution. It's a brown revolution, because it's been destroying soil. And when you have no biology left in your soil, there's no way to grow good plants. Get that biology back into the soil, and now you can have a healthy plant that is never, ever stressed for nutrients. Every second of every day, your plant should be fed by this nutrient cycling process going around the root system. You can think of that layer of beneficial organisms around that root as a castle wall, preventing diseases from being able to get into your plant. Not even the plant doesn't, or the bad guy, the disease-causing pests and parasites don't even know your plant is there if you've got a good, healthy set of organisms. Structure is built by the microorganisms. There's a whole list of 5 different. I'm getting feedback. Somebody's talking.
Yeah, I'm getting feedback too.
I think that's Wayne in the background. Wayne, could you mute your mic, please?
Sorry, Wayne. So we go through an explanation of all these overarching principles that apply every place on this planet to how plants and microorganisms in the soil interact to make the plant healthy, to allow the root systems of the plants to go deep down into the soil. If you've ever been told that your root system only goes down just a few inches, just laugh at that person. They have no comprehension of the reality of what soil is. They're playing with dirt, and you can't grow good plants. You can't grow healthy plants in dirt. So at Colorado State University, working on documenting, quantifying, putting out the ecological monograph that was published back in 1985, which is pretty much the first time anybody delved into how the organisms in the soil and the plant interact with each other. As I traveled to the rest of my career, finished up my PhD in 1981, postdoced at Colorado State University at the Natural Resource Ecology Lab for about another 5 years. After that, doing a lot of looking at what is the food web in different parts of the world, different stages of succession. We all know that, a big disturbance is going to result in absolutely no plants growing, no nutrients present in that soil, all kinds of, that's dirt, doesn't have any good biology in it. There's a very important definition of what dirt is. Extremely low sets of microorganisms, or only the bad guy organisms, strictly bacterial dominated, and only the problem bacteria present. But then nature is going to send weeds in there to get some foods, get the exudates started into that soil. So now the bacteria and a little bit fungi, as those weeds grow, they're putting in more fungal foods. And so now you're seeing that fungal component starting to increase. So you're going from bacterial dominated, almost no fungi. Fungi are going to creep up a little bit. Bacteria typically stay in about the same place. So over the course of succession, through vegetables, through coal, kale crops, those kinds of things, into veggies, into herbs and forbs, grasslands, highly productive grasslands, into our agricultural systems where we're growing plants like corn and wheat and barley. Equal biomass of fungi to bacteria is required to set the stage to select for the growth. Well, why should this be? Well, you're going to have to go take the foundation courses, because it's another fairly long explanation for why this equal biomass of fungi and bacteria is so important if you want to grow the most productive pastures. Then, of course, in the real world, nature keeps going through succession, and pretty soon the fungi are going to be predominant, and that's where you grow shrubs and vines and grapevines and blueberries and blackberries and things. Go a little bit more fungal, and now you're into deciduous trees. So here's all your orchard crops. Go a little bit further, a lot further, and you're into conifers and old growth forests, where 75% of the volume or the weight of a gram of soil is fungus. It's not sand, silt, clay. It's fungus that makes up the majority of the soil when we go into those highly stable, highly carbon sequestering systems. And you have to understand that the sequestration are fungi. It's fungi that do that work, not bacteria. You want to get rid of carbon and blow it off into the atmosphere as CO2? Yeah, grow some bacteria. And that's explained why it works that way. So from Colorado State University, I went to the University of Georgia, where we first started managing biology in the soil and showed massive increases in the growth of corn, soybean, alfalfa, lots of those kinds of agricultural crops on large scales. Lots of people still working down in that area on that work, Horseshoe Bend Agroecosystem, for example. From the University of Georgia, I came to Oregon State University, and along with my husband, we both got professorships here at Oregon State University. And of course, my husband is still a professor of nematology at Oregon State University, which explains how I know so much about nematodes. But I did some work on genetically engineered organisms where headbutting. We showed that the genetically engineered organisms that we worked with were extremely dangerous. If that had been released out into the real world, we would have lost terrestrial plants because of the changes that would occur in the soil and the way nutrients were cycled, and the consequence of the compounds made by that genetically engineered microorganism, which originally came from soil. The parent organism is extremely beneficial for plant growth. The genetically engineered organism would kill all plants. It might affect your life just a little bit to have that released out into the real world. Luckily, we came within 2 weeks of it being released into the real world, but we managed to stop that release because we had the data to show how dangerous that genetically engineered microorganism actually was. So I do sometimes say I've saved the world at least once, probably more like 2 or 3. So from there, I had a huge tussle with Monsanto, and of course, Monsanto won. Monsanto puts millions, $75 million dollars into the budget of the universities, and they put money into every university, well, I'm sure you all know this, so I don't have to say that. But in my particular case, it was a simple business assessment. I only brought in the best years, I only brought $2 million into the university. Monsanto was giving 75 million. So think about that for about a microsecond. Business people, who are they going to choose? So I could see the handwriting on the wall. I started Soil Food Web in 1996, and it's just been growing ever since. There have been a few years of frustrations where people just didn't want to listen, they didn't want to believe. But we work with probably over 10 million acres and in agricultural work around the world. Most people have heard about the Soil Food Web and possibly about myself as well. And we'd like you to come and learn all the, if you will, secrets. I love it when somebody says they're secrets. Well, they're not secrets. We're going to teach you about them. It takes a little time and effort, but I don't expect you to know chemistry. I don't expect you to have a background in those. So we make certain to explain all the background so you can understand. So it's not like going to an organic chemistry course where, whoa, that's hard to figure out just because they go so fast and assume so much about your background. We don't assume that. And so hopefully it's all clear. And if it's not clear to you, if you are being confused by any of the concepts or any of the chemistry, you ask questions, and we have people here to help you understand that, take you through and make sure that you do understand it so you can keep going. So I mostly these days work with clients, answer questions, make certain that the online courses are kept up to date, where all the latest and greatest information is being added to the foundation courses. It's added into the consultant training program. So hope that wasn't too boring. Back over to you, Brian. I think we're probably ready to start answering some questions.
I think we are ready to start answering some questions. So we moved into the Q&A, and thank you, Elaine, for that great introduction. So let's start with a question from Benjamin, and he asks, is it possible to farm without using any chemicals? Great question, Benjamin. Who on our panel wants to tackle this one first? Elaine does. All right. I'm sure you have something to say about this.
I have something to say about that, because that's what we do for people. We get you off of pesticides and inorganic fertilizers, everything with the toxic chemicals. Well, guess what? They're toxic and they kill things. And that's really a Newcomb approach to dealing with growing plants. Let's sterilize everything, except if you sterilize your soil, it's not soil anymore, it's dirt. So yes, we teach you how to grow plants. And remember, you've got to know about what the proper balances are as you go through succession. What is the balance where the plant you want to grow is going to be selected for, and every other kind of plant is going to be selected against. So we got to get you understanding that process as you go through succession. And then you need to be able to have the criteria to decide, well, yes, this is in that group of plants that requires 5 times more fungi than bacteria. So you set up the right set of organisms. Or maybe you're trying to grow sweet peas, and you really need a fungal to bacterial biomass ratio that's down about 0.5 or maybe even 0.3, somewhere in that range. Maybe you're trying to grow, fill in the blank. And we will help you understand how to figure out for yourself. Because see, we want you to be able to do this without somebody else behind you, like manipulating you like a puppet. You're farming should require people to be very smart and intelligent and clever about paying attention to their plants and getting the biology right. It's not really that difficult. It's not rocket science, really, but you do have to pay attention. You do have to be willing to take some guidance from Mother Nature, because she's been sending us messages all along and we've been ignoring them. And if you ignore Mother Nature, what happens? Who's going to win this war? Is it human beings or nature? And we're seeing that in climate change. And everyone who wants to be ostrich with their head in the sand, they're not going to be with us for too much longer. So, did I answer that question?
You answered it great. I have something I want to add to it as well. So here on our homestead, this would be our 9th year without using any chemical inputs at all. And we have, in fact, a really good closed loop system. Our homestead here produces enough materials for me to make compost to be able to use back into my system. And I would say that over the 9-year period of time, every year I get more productive. I get more growth, and I'm doing less management. Once I've got my food forest set up and I've got my orchard systems going, I spray with compost tea a couple times a year, and then manage some of the herbaceous plants I have underneath and do some chop and drop, and just typical pruning and thinning of the fruit. And I tell you what, it's amazing the amount of production that I get out of my homestead here now. Way more than when I was putting chemical inputs. I mean, way more. So that's my own personal experience as far as a long-term, hey, without chemicals, it just gets better as you get that soil biology set right for the systems that you're trying to grow.
So do anyone else have stories to contribute?
All right, Keisha.
Come on, Wes.
I would just say that whenever we are growing our food biologically, the taste, the flavor is so much different than what you pick up from the grocery store. I mean, even the organic produce is lacking compared to really diverse, nice soil. The food that comes out of that is just sweet and tasty. The flavor profiles are insane. It's just a funner food to eat, more enjoyable.
Agreed. All right, how about you, Wes?
Well, I'm still a lab tech. Of course, I'm not as far along as Brian and some of the other consultants, but I have had the experience of seeing a pest show up mid-season on my vegetables, aphids, and being able to get rid of those pretty quick and then go back to compost tea applications for the rest of the season, and never saw another problem for the rest of the season, and finished the season with plants that were so healthy, like I had never seen that before. And that's my experience so far. That was my life-changing experience. It's really just astounding, the difference.
Nancy, Tanya, anything you guys want to add?
Well, I'd like to say, I'm in the Netherlands, and I work on occasion for the botanical garden, and in the greenery they used to use chemicals, but when I treated one of their coffee plants because it was overrun with the mealybugs, and proved to them that if you treat them with compost tea and in the soil compost extract, I combated the problem for them. And it was impressive enough to them that they have never bought any chemicals again. It's really good, yeah. But it doesn't. Yeah, right now the coffee plant is starting to bloom, flower, and it's showing lots of little beans. So let's see how it develops. And there are lots of coffee beans on it right now. And it took, I think, in the last 10 years, it didn't produce any coffee beans. But so I can't weigh it against a previous harvest or anything like that. But it's great that it is growing coffee beans right now. Yeah.
Awesome. How about you, Nancy? Anything, Dad?
I don't have anything big to offer because you guys have all said it. But but growing without chemicals and fertilizers and stuff is amazing. And the life you see in your garden or wherever farm, wherever you're growing, is incredible. And and putting in a lot of diversity of plants, then you've got places for the predators to hide. And so if there is ever an issue, the predators come in as well. And it all starts with a healthy soil, which grows healthy plants, and then the whole ecosystem works for you.
Yeah.
Exactly. All right. Thank you, panelists. Okay, we got another question here. This one's from Alicia. Alicia asks, any thoughts on how knowledge of the soil food web relates to urban farming? All right, you may want to take an urban farming question. All right, don't be shy, panelists.
I can start just quickly to get the ball rolling. Is
sure.
Yes, because I'm in more of the suburban urban interface, and actually I just went to a no-till growers thing here down in San Diego, which was wonderful. And this guy had a whole urban farm where he was, had a whole business around it. The soil food web principles apply no matter where you are. I mean, we talk mostly about farms because they really need it because they're so big. They're going to feed huge groups of people, but I say let's get everybody in the urban and suburban interface doing it as well, because it's just healthier in the long run for our planet and for us. If you're growing food, of course, but even if you're growing other plants, why are we putting all these poisons out, which will just run off and go into the underground water? So yeah, all these principles apply big time in an urban farm setting. So use it.
Even if it's not an urban farm, I mean, what is a grass lawn than a meadow? Or it's if you think of it, any plant you're growing anywhere in the world, you could use biology to grow it, and it'll be better, healthier for everybody who's involved.
Yeah, and there's a lot of people too. I mean, I have a lot of urban clients. Either they grow their own food or just for their own yards, and they just don't want the toxic chemicals put on their lawns. Their kids go out there and play. There's this kind of an awakening of, wow, okay, yeah, these things are pretty bad. When you get a bottle of it and it has all these warnings, could cause death or something like that, you gotta ask yourself, why am I putting these on things I eat? But, yeah,
it's like when you think about the chemicals, and you have to put up these signs that say no entry for 2 days because that toxic chemical has been sprayed. When you go out there and you put biology on, and you either do a application of extract onto the soil or foliar application with a tea, we really should be putting signs out there that says, please come roll around in this for a while, because you're re-inoculating all of your surfaces with these beneficial organisms that you need to have in order to protect yourself from diseases and problems. You ought to be drinking some of this stuff because it replenishes the biology in your digestive system, those organisms that you need to have in order to digest your food correctly. Kind of our stomachs, our intestines aren't a whole lot different than plant roots. We're putting out exudates to grow the correct sets of microorganisms, but where did the correct sets of organisms come from? And if you've been eating food that has preservatives in it, if you've been eating food that has toxic material in it, you've destroyed that biology. How do you get it back? You've got to be eating things that have these beneficial organisms on them. And why not be making your own compost so you know exactly how that compost was made and what was in it, what is in it. You use your microscope to make certain that it's all the beneficials and none of the bad guys have shown up. And now you make the extracts, you make the teas, you put the compost out. Everything you're putting out is beneficial. You go stick your hands into that compost pile and you get all those organisms on your hands, and please then go eat French fries and lick your fingers.
After you use your microscope.
Yeah.
I was going to say, I have one more urban story, because I just recently helped my sister who had a big rose bed, and she said, my soil is terrible. Can you help me? And I went, yes. So, we went out and we got compost. We got compost tea. And then I told her, which she just was appalled. I said, call an arborist and please get some arbor chips in here. So we did all that, and she could this is so much. What am I going to do with it all? I said, you'll use it over time. But anyway, so she shoveled and got it all done. And she's particularly said this one place in her garden would never grow anything. Anyway, she recently sent me a picture, and the place that was the worst is just full of life with her flowers, and it's spring, so she's just thrilled. I said, just keep it up. So Anyway, yeah, so no matter what situation, yes, the soil food web can help.
For sure. Okay, anything for the other panelists?
Nope, it's
All
fine.
right, I have a dovetail question from Leslie. It goes right into this, which is how can this be applied to gardening in containers? And for sure, even on our own homestead, we've got buckets, containers, and things like that that we use, and it works fantastically well. You can just aquaponics, hydroponics. There's so many different, applications of managing your soil food web. That yes, containers are a great application, especially for those that have very small yards or live in apartments that just have a balcony. Anything else, panelists?
I was going to say we've had some like small potted plants that we use 100% pure compost, and it wasn't like a potting soil mix or anything like that. And what we found is that you actually have a hard time keeping the moisture. You feel like you want to water it more than you need to because the compost holds moisture so well that you end up actually overwatering some of the plants, because it's just holding onto it like a sponge. So you got to actually train yourself to back off. You don't necessarily need water every single day.
Yeah.
Yeah, when we've worked with grape growers in very dry parts of Australia or the United States, or well, anywhere else, we have reduced water use by 70%, which means that they now have enough water to double or triple the amount of grapevines. So, it's definitely a benefit in anywhere where we're in a water shortage area. When you have summers that you have drought late in the growing season, if you've got the biology in the soil, you've got the structure, you've stored the water in the soil, and your root system should be able to reach down to that water. So we hugely reduce the requirement for watering things.
All right. Okay, I'd say we move on to another question here. This one's from Rowdy. And Rowdy says, I've had difficulty getting a proper compost pile started. Is there a good measurable method for measuring the components to make a pile? For example, 10 5-gallon buckets per ? All right, who wants to tackle this one?
It's very
There we
clever.
go, the composters. Yeah.
Yeah, whatever
And that
measuring you use. You could, I like to use those like cheap totes that you can pick up at Walmart. I think they're like storage bins. They're like 14 gallons. You just want to make sure that you have enough material to start out with. When you're using 5-gallon buckets, your pile might be a little small. I would maybe use 25-gallon buckets or 10 of those big 14-gallon totes or more. It can be hard to maintain temperatures, especially in the winter when your pile is so little. You need enough mass there to hold onto that heat.
Yeah, and we go over these requirements. We talk about recipe and what are the components, and you have to pay attention to what the animals were being fed. If you're using manure, you've got to pay attention to what toxic chemicals were applied, what inorganic fertilizers and pesticides were applied. And you're either going to have to get those microorganisms that will be able to deal with that toxic chemical, or you don't use it. So there's requirements like that. And so the compost foundation course goes through all of that information so that you can make good compost right away. And of course, we make compost in 21 days. It's not a 2-year process. It's not a 6-month process. It's if you want to make compost rapidly, we have those methods and they're recorded. You just need to come and take the foundation courses.
Yeah, and that's that's a nice thing about the courses too, because when you're in that kind of process, you need to be able to assess materials correctly. So we teach you how to do that. And there's a recipe, it's
got to monitor temperature. You have to monitor water. There's all these things, and we teach you hopefully how to pay attention and measure the things that need to be measured, and not just measure stuff for the joy of measuring it.
Yeah.
Teach you how to make your own recipe, because one standard recipe doesn't work all over the planet.
Yeah.
You gotta choose for what we have available as far as ingredients go. So.
Yeah. All right. Anything else from the panelists before we move on? Okay, we've got other Oh, go ahead, Elaine.
I saw a question there that just came up. They ask if there's a maturing age stage after the 21 days. No, the 21 days includes the maturation, maturation being coming back down to ambient conditions. So you have a completely finished pile in 21 days if you're following directions, if you're doing it right. We've got those microorganisms well trained.
Yeah.
Yes.
We have that photo of our compost piles, and there's 4 of them, and I think they're 3 days apart each pile, and it's like 11 days or something throughout each pile. And the first one just looks like wood chips and grass and everything mixed together, and it's slowly the next one's kind of a tan color, then it's like brown, and then the last one is like almost completely, it looks like finished compost, you know, it's just that rich cacao color. And it's only 11 days,
Yeah.
you know, it's 11 days in. It's still not even done coming to temperature, so.
And when the microorganisms are there, they process the organic materials so quickly. It's funny to me when people say, like, you know, wood chips won't be broken down quickly. If you've got your fungal component, they are gone. Like, it doesn't take very long at all to be obliterated.
Turned into soil.
Yummy stuff.
We're soil makers.
Yep.
Exactly.
Microfarmer.
Well, on the theme of compost, we got another compost question here. This one's from Jasmine, and Jasmine asks, I live in an extreme cold climate, 65 frost-free days. What is the best way to store my compost over winter? Is it inefficient to apply it in the field before the snow covers the ground? Okay, who wants to take that one?
Fungus actually likes to grow in the cold, so applying your microbes right before it snows, I think, is the best way to go about it. We find that when we're applying the microbes in the fall, before it gets really cold and that snow layer hits, that once spring does come around, which sounds like it's a long time for you, it's really glorious what happens over that winter time. You wouldn't imagine that fungus is growing like crazy under the blanket of snow, but it actually is. And the other microorganisms just go to sleep. They don't die or freeze to death. They wake up as soon as the conditions are appropriate for them. So even where it's very cold, I would say not a waste of time at all.
Yep, and you have to understand that as long as you've got a buffering, you have a layer of snow or a layer of mulch, for example, there's going to be free water all the way through the wintertime period. At that interface between the snow and the organic matter and the soil. And it is in that free water that the cold loving fungi and bacteria, some protozoa are still in there. They're just going gangbusters in the winter under the snow. The highest rate of decomposition is recorded from litter bags where they're in that position at that interface of the snow layer and the snow. So we were, the team I worked with at Colorado State University, we were the first ones to report that phenomenon. And most of everybody else said, you're crazy, you don't know what you're talking about, you know, this is ridiculous. And then several other places in Colorado, up in the mountains on the East Coast as well, repeated those experiments and it showed exactly the same thing. The highest rates of decomposition ever recorded on this planet are in the winter under the snow. So get your organisms out there, get them established in your soil. Now let it become winter time. And they just keep working for you all winter long, building structure, holding on to nutrients, all the great things that they do, waiting for your plant to start telling them exactly what they need to do to keep the plant happy.
Great. Anything else from other panelists?
Oh, that's fine.
Good question, Sandra. Okay, so let's move on to another question from Stefan. And he asks, oh, I'm sorry, no, this question is from Sandra. My apologies. Sandra asks, I tried starting fava beans last fall, and the problem soil, even with a compost base, has failed to bring them up. It seems this garden soil here is dead. I've been working on it since last fall. How should I take, how long should it take to life? Okay, who wants to tackle that one?
What
How do you
kind of?
know? Yeah, how do you know if something's live or dead? What's our measuring tool?
Got to use a microscope.
Yeah.
Yeah.
Yep. So, Sandra, the best thing to do, I would suggest is get a soil sample done, have a biological assessment to see exactly how dead the soil is, and then if you apply good biology and your management practices, are going to be there so you can support that biology, not spraying chemicals or tilling things like that. You can within 1 year take extremely problematic, horrible soil and make it very productive. It all comes down to, do you have a good source of good biology? Is your management practices, supporting that biology once you place them into the system, and you can turn around in a year's time frame.
Often you may see it in a few weeks, though.
Sure.
It might not be perfect, but you never. I mean, often we get reports back of people adding a compost extract or compost to their, their problem zones, and just seeing, amazing results within a couple of weeks. But then, the following year, you're seeing what the plant can really do. And then as the years continue to build, like you were saying earlier, Brian, the production just goes up and up and up until it's really blowing your mind. But you might be able to fix that little area quickly, as long as it's still moist where you're at and not too hot and dry.
Yeah, and you also said that you put compost out, and you got to make sure you're checking your compost wherever you're getting it from. If it's municipal waste from the city, it's probably not that beneficial. So, got to get on the microscope. You got to look at the compost that you're buying and what you're using to know that is it even worth your time putting that compost out into your field.
Yeah. And that's a good point about the compost quality. I just looked at one of my clients sent me a sample of compost, and the compost company told them this is their fungal mix. This is it's got a lot of good fungus. Well, when I looked in the microscope, it had 2 organisms: it had bacteria and disease-causing fungi. So if they wanted it as a fungal mix, it was just the wrong fungus that was in there. So you ask, if I would ask every compost producer, what is, have you done a biological assessment? What's the biological profile of the compost that you're selling? If they just look at you and go, huh? Biology what?
A lot of companies nowadays love the terminology. It's like microbes are hot right now. So it's like that's what it's on everybody's packaging. It's everywhere, but you can check by putting it under a microscope, and you can actually see for yourself if that's actually what's going on.
You can certainly send around samples to somebody, who's taking these courses, who's got their lab, and, just go to all these fungal producing compost companies, ask for samples and send it out, and then show them your report and see what they have to say. Perhaps they'll change their practices if you,
call them out.
Yep. And you've got 2 people right here. So Nancy and Wes, that's what they do. They take samples from people and send you back a report telling you what's in it. Yeah, are you playing with dirt, or are you actually playing with soil? And the difference is the biology.
Yep.
I wanted to add one more thing, and that is one of our practices is that we coat seed. We coat the seeds with the microorganisms. So if you have a very bad soil, as long as they germinate and they start their photosynthesis, they will start to grow the microorganisms as soon as they get above ground. So one of the practices is to put a coating of microorganisms around your seeds before you plant them. Well, you'll learn it in the course.
Yeah, that's a very effective way to do it. Yeah, I use that practice all the time. It's great. Okay, anything else from other panelists? Okay, now we have the question from Stefan. And Stefan asked the question, at which time of year should you apply compost tea? End of dry season or winter or already during the dry season, etc. All right, who wants to take that question?
Yeah, I'll take it.
All right, Tanya.
Actually, compost tea, you use compost tea for specific reasons. You want to protect the above ground growth of your plants. You want to protect them with the microbes. So that's why you spray it with compost teas. But when do you spray it? So when the branches are without leaves, of course you will not spray anything. But once how do you call them? The green things that grow on blank.
Buds.
Oh, God. I had a blank. The leaves that come out, you want to spray it. And also once they start to make the fruit or the end product, you also want to quickly give it a once over, because once in the smallest part of it, it will grow with the fruit or whatever you're growing. The microbes will grow with it. So you apply it at specific times, not all year round, or when it's dry. That's not an issue. That's not a criteria.
Yeah, it's not a good idea to be putting the compost tea on when the plant's already starting into dormant stage.
Right.
When the leaves are starting to turn brown, there's not much point or purpose. But certainly at bud swell and then bud break, when we're dealing with perennial plants, or at the first true leaf stage, as an annual is starting to grow, you want to get that protection on those leaves, and then those organisms will continue to grow and stick with it. So we have very specific recommendations for the times to be applying, but it depends on all these other things that might be happening in your system. So it's take the foundation courses so you can find out what all these different factors are that you want to pay attention to, and then that's really what's governing the reasons for you to apply the tea.
Great. Any other panelists want to weigh in?
No, I'd love to have a microscope question though.
Yeah, exactly. None yet. I'll keep my eye out for them. Okay, we got another question from Steve, and this is an interesting question. I'm curious to see what our panelists have for this one. Is there a probiotic product available with these microorganisms that nutritional consultants can provide to their clients? I have a kind of a weighted answer, but anybody else want to weigh in before I say it?
Are you talking about food grown in healthy soil, Brian?
I was just going to say, just eat the fruit. In biological systems, there's your probiotic, right? So if you're spraying with compost teas and you're covering the outside of your fruits and vegetables and with all these great organisms, then eat those fruits and vegetables. And from a nutritional consultant standpoint, it seems like that would fit right in highly nutritional dense food with those organisms already on them. Seems like a winner to me. Anybody
I don't
else?
know, Brian, though. I've never actually checked it out, but on my social media feed, they know that I'm into microbes, and they've started to advertise to me all of these different soil. They're probiotics, but they're made of microbes that they've isolated and grown in a lab that also grow in the soil. So it is a thing now. People are starting to create probiotics for humans to eat that are
soil-born,
of the soil, so to speak, but still lab grown. So it's going in that direction, Facebook says.
It's just, it comes to the point of like, how possible is it to capture all of these creatures that exist outside, and put it into a pill form? There's such a vast variety of microorganisms that live outside that you're not going to be able to bottle them all up. And are these 6 that they've isolated, is that necessarily the best, or something that you definitely need? It's hard to say. So the best is have it come naturally from outside.
Because when you look at what happens to microorganisms when you grow them in a really rich medium, they don't have to work at all for their nutrients. They just, couch potatoes are what we call them in the trade. You're turning all those microorganisms that should be doing specific jobs, you're turning them into couch potatoes where they just have to lie there and, suck up the nutrients. So are they really the sub portion of a population that are going to work really well in your digestive system? They're not going to be able to tolerate the competition that ought to be going on in your digestive system. So, and 6 species. How many species of bacteria or fungi or protozoa or nematodes should be present in your digestive system? And it's probably upwards of somewhere around 75,000 species of bacteria, 25,000 species of fungi, 1,000 different species of protozoa, and maybe only, 10 or 20 species of nematodes. So, starting to understand what all these organisms are doing in your digestive system, that's kind of one of those places we need to delve into. And which organisms are alive and happy and doing their thing in which micrometer of your digestive system. What's the community you need to have, okay, so see where we've only begun trying to understand those things. We've only begun trying to understand the biology in soil as well as on the surfaces of plants. So lots more interesting things to come along. But when you realize the diversity that you need to have in your digestive system, eating a pill that's got 2 or 6 different species of bacteria, are you really making even the slightest dent in what you need to have? Probably not. Go out and eat plant material that comes from a system that doesn't have pesticides, doesn't have inorganic fertilizers being applied, because all of those things, all the pesticides, all the inorganic fertilizers kill those organisms.
It is interesting when before we started Catalyst and started making compost, my digestive system was just wrecked. I wasn't processing any food for years. I was having so many problems with it, trying every probiotic on the market. I've been to the doctor so many times and no one could tell me what was wrong. And it was only, weeks, maybe 6 or 8 weeks after we started making compost that it just was cured. I have 0 digestive issues anymore. I didn't eat the compost, but like Elaine says, I go out and I check the moisture every day. I touch every pile. And then from my hands, it'll get on the pitchforks and it gets on my doorknobs and all over my steering wheel of my car, and eventually makes it into my mouth. And I actually don't get to eat a lot of plant material from what's grown in our compost because we sell it all. But just being near the compost and touching the compost and living with the compost has literally cured me. I can't imagine what else it would have been that would have just snapped me into full repair like that. So there's no science behind it, but it worked for me.
Yeah.
We need to have science on it, but who are we dealing with? We're dealing with big pharmaceutical companies, big chemical companies, and but they've got the money
for us to do microbial research on how they could stop making money. Oh, what a shocker.
There's one scientific paper out there that's really interesting, and they found the MST bacteria that grows in soil, but is really very much present in compost. And one of the characteristics of this little bacteria is that it gives off molecules that stimulate our serotonin levels, serotonin. So you get to be really happy when you turn your compost.
That's great.
That's why we're so smiley.
Exactly. We're happy.
It's not that we love our jobs, it's the micro-protecting the serotonin in our brains. They're forcing us to make compost.
Like drugs.
The bacteria are in control.
Exactly.
They are managing us.
I don't know if anyone's ever looked into kombucha as far as all this is concerned. I've looked at, a sample of a beverage product under my microscope, and as far as I can tell you, just from what I see morphologically, it looked healthy. But as far as how many species are in there, I don't know much about the production process for kombucha products. If it's anything like, your average soil inoculant product, then it's going to be problematic because, only such an infinitesimal number of bacteria or other organisms that can be packaged and survive that whole process. But that would be an interesting thing to look into, I suppose.
I heard.
Question: when I look, I see a lot of yeast, which, bacteria and yeast, that's what it stands for, right? So I kind of imagine that maybe it's not the health drink that it is expensive. And so I can see why they would push it as like a super tonic, and it's tasty. I think kombucha is a great way to preserve, a drink, but it's a lot of yeast when you look at it under the microscope, a lot of yeast. So
yeah.
A lot of lactobacillus typically as well. Limited diversity.
Yeah.
I heard scobies can get upward to 100 different species of yeast and bacteria. But again, that wasn't any kind of scientific research. That was just something I read about. So who knows? Take that with an extremely grain of salt. Yeah. Okay. That was a great question. I think we'll move on. Tanya, you got your wish. The microscope question is coming in. This question comes from Brian. So I like that name. It's got 2 Brians in it, which is fantastic. All right, so here's the microscope question. What type required for this type of, what type of microscope is required for this type of work? And does anyone have a favorite brand? Okay. Who wants to start with this one?
I
All
can
right,
jump
Nancy.
in just quickly because I've got the probably the lowest end, or maybe Wes and I might have that same. Anyway, I have an Omax, and it was low end. It was what was recommended in 2016 when I took the microscope class, and it's binocular with a built-in camera, and it has been wonderful. I've had a great journey with it. It did come with software for the camera originally that I had on a PC, and the software was not that easy to work with, but when it worked well, it was fine. And then my PC crashed and I ended up getting a Mac. And so now I just use a USB cord to connect it to my computer and use photo booths. And I get amazing pictures and videos of the microbial life. So again, it's very basic, and it has done well by me.
I think we still recommend the Omax. So go ahead, Wes.
I was just gonna say, I think Nancy and I do have that same microscope, and I've been using it for 2 years now, 2 years plus. And you get to the point where you start going, maybe some bells and whistles, and maybe what do you call it, the objective that is not a plan objective, so that things are just a little more clear, and comfort, things like that. But yeah, it's a great microscope.
And it's not very expensive. What did you pay for yours, Wes?
400 and change.
And mine was $350.
Yep. So not that expensive to get up and get going, get your own lab up and going. Now you can start charging for the samples, and pretty soon you've got enough money accumulated to buy something fancier if you so desire. So like Keisha, you just bought a fancy schmancy microscope because you are getting better pictures.
Yeah, I'm getting better pictures and better video. And what I really like about the scope that I have, which is the LW Scientific, is that there's a lot of really neat little bells and whistles that I didn't even know existed before. Like, say that I have a group of 20 people in the field and I'm assessing something on my microscope. My microscope can actually send the photo out to their cell phone. And so they can look at their own phone and see what I'm doing on my microscope. And then I can also take my microscope and plug it into one of the new 4K televisions, and it'll blow up on the big screen exactly what I'm looking at for the convention. So for the work that I do, I'm really into the show and tell side of the microbe stuff. I really play around with that a lot. So I actually saved up all last year and I got this whole package. But my scope also comes with an epifluorescent unit. So now I can look at what's going on the leaf surfaces. It does a lot of things, and I chose it because it's specifically suited to the work I do. I also have like 2 or 3 Omax microscopes that we keep, and those are great too. You can take great photos with those and do all your counts and everything necessary with just the simple guy. So.
Yeah, I'm glad you brought up the epifluorescent because in my lab I run 2 microscopes. One is the Omax I got way back when I started, and I do most of my biological assessments just on that scope, and that's the scope I use to travel with as well. When I'm going to my client locations. And then an epifluorescent microscope. So again, I could do leaf surface analysis of colonization. We talked about foliar applications of compost tea. Well, if we're going to go out there with my vineyard clients, I want to make sure that I got a good coverage on the surface of the leaf. I can pull that leaf in and see what kind of coverage do I have, and the epifluorescent will give me that information. I can also look at mycorrhizal root colonization too with the epifluorescent microscope. So yeah, it doesn't mean you have to have an epifluorescent microscope, but it's another tool that you have. And some of your clients are going to want that or actually require that from you.
It's really fun.
Oh, it's fantastic. I love looking through the epifluorescent. It's really cool.
I know. Just the normal microscope sucks you in, and you say, I'm gonna get this sample done in 10 minutes. And you start working on it, and 3 hours later you go, oh my God, what happened to the time?
So many of my samples are in photo shoots.
Yep. People have asked about, like research papers or information, specific information. Somebody was asking for information about these microscopes. Where do you order them? Where do you get them? The website, please. Go looking on the website, or, to get the specific information, you might want to take the foundation classes, because all of this information is in there.
Yeah, and that's what the foundation course 4, which is the microscopy module, really breaks it down. So we're looking for a shadowing microscope, needs to have an Abbott condenser, an iris diaphragm, all those kind of things. So you'll get introduced to those terms and what that equipment actually does, and then also tuning them to make sure that you can shadow effectively, so you can see your microorganisms. Yeah. But once you learn how to use that tool, oh my gosh, it's fantastic. You really want to know something? Just go take a soil sample and go look into the microscope. Go, okay.
What's going on in my soil? Well,
exactly.
You've got the tool, and it you can just put a sample on really quick and get a general idea of, wow, too many bacteria, not enough bacteria. Fungi look pretty good, or whoops, no fungi home at all. And now you know what's going on in your soil and what you need to fix.
Yeah, and one other thing I've mentioned too is is becoming a trend for me is I'm getting a lot of clients that are just farmers, right? They're running their operation that want to learn the microscope. They want to use that as their own diagnostic tool, so they can do either qualitative analysis, which is just saying, hey, I'm going to pull in a sample and make it just a subjective assessment by looking at it, or they want to do their own quantitatives where they're actually doing counts and things like that. So, and I encourage it. I'm happy to see that my clients want to have a microscope and use it, because it's just a tool that they can use. It just helps them in their operation. All right, anything else from panelists before we move on to the next question?
Yeah, I want to say that I took the list of recommendations of what the microscope should be, because we don't, in the Netherlands, we didn't have the OMAX. So I went to my microscope man and went through all the requirements, and I bought an iScope with a high-speed camera, and I'm so in love with my high-speed camera, because when you chase nematodes, they wiggle around like crazy. And you really want to capture them because they're so wonderful to capture. Then a high-speed camera, really fantastic, yeah. And
We need to get that information from you and put it into the FC, especially for the European students. Yep.
Sure. Yeah. Very happy.
It's a nice. I think someone brought one of those to one of our microscope in-person classes, Elaine. It was a really nice microscope.
Yep.
Yeah. It's portable, so I also take them to clients. When I went to Belgium to the farmer, and I hooked up his TV on the microscope. Yeah, it has a HDMI,
HDMI.
Yeah,
HDMI.
it has a special cable.
And I think Eva has that microscope.
Yeah,
she's using it.
Okay, we're getting close to the top of the hour. I say, Elaine, we'll tackle one more question.
Yep, sounds good.
Okay, so this question is from Merlin, and Merlin asks, a farmer told me that he had to reseed his alfalfa every 4 to 5 years due to N buildup. Anybody want to take that one?
That's something that growers get told about all the time, that nitrogen builds up when you're dealing with nitrogen fixing plants, and the data for that is really questionable. What I see happening more often is that alfalfa produces compounds that prevent their neighboring alfalfa plant from being able to grow into their root system. So allelopathic compound is what you would call it. And if you don't have the right biology in your soil to chew up those allelopathic compounds, those allelopathic compounds continue to accumulate. And at about year 3 in your alfalfa production in that field, that allelopathic compound starts affecting the alfalfa itself. Because when the alfalfa starts to regrow in the springtime, And those root systems are now growing into soil that has really high levels of that allelopathic compound. The plant can't grow because of that accumulated compound that it put into the soil in previous years. You're supposed to have all this good biology in your soil. It's supposed to be decomposed through that winter time period, but because we've destroyed that biology in the soil, it doesn't get decomposed. And therefore, you have to go out there and manage your field to encourage the bacteria that are in your soil, hopefully, that at least you hopefully have as a remnant, and that you've got species of bacteria that can decompose that. Yes, it is a compound that contains a fair amount of nitrogen in it, but it's not the nitrogen that's causing the problem. It's the allelopathy that is the problem. If you maintain really good biology, you're out there and you put in your compost and extracts and teas, and now you're maintaining your biology at the right balances, you never have that problem. The allelopathic compound is going to be used by the microorganisms as a food resource. And so you don't have that problem. Anyone else run into this in-serve kind of the nitrogen problem in alfalfa or other other nitrogen fixing plants?
No, I haven't come across that.
Anyone else?
No,
You win?
never.
You win? All right. Well, I think that will conclude. I think that's the questions that we're going to go through today. So I'd like to start off by thanking Wayne and Mark, our hosts, for allowing us to be able to share this information on their platform. Thank you so much. Also like to thank all of our panelists that have been providing such great commentary and dialogue and answering questions from the community out here. And also I'd like to thank Luke. He's been helping us behind the scenes, him and Mark both, of making sure this webinar runs smoothly. And I'd like to thank all of you for attending. This is great. We're able to share this information with you. And always thank you, Dr. Elaine Ingham, for doing what you do. I really appreciate it. So, Elaine, anything you want to close with?
Oh, that sounds pretty good. It's we're here at the Soil Food Web School, hoping that you'll come and work with us. We'll be able to give you the training that you want, so you can go out and make your own lab, and hopefully eventually become a consultant and have clients that you work with, and you can help us convert agriculture into something that's strictly biological.
Yes.
Agreed. Okay. Thank you all.
Bye all.
Real quickly, this is Wayne.
Hey, wait.
Goodbye, everybody. Again, Elaine, thank you so much.
Thank you, Wayne.
Thank you, all the panelists. And check this course out, everybody. There will be replay available soon, and the chat roll, the question roll will be with it.
Great. Thanks, Wayne.
Bye.
All right. Thanks, everybody. Bye.
Bye. Take care. Enjoy.